So we're going to shift over to RA a little bit before the break. I always talk about lupus research. It's like going into Afghanistan. You get in, you never get out. I am a clinical trialist, so we're fortunate to have not only a phase 2 through 4 unit, but we have a phase 1 unit where we can put people in for several days overnight and have multiple beds. So we get introduced to a lot of molecules in our different rheumatic diseases very early. And it's nice, at least in rheumatoid arthritis, that we have endpoints that have been validated throughout my career that we feel comfortable with. It's such a difficult thing with heterogeneous diseases such as lupus and Sjogren's. So yesterday I showed no disclosures. Today I showed the multitude of disclosures. We have a staff of about 30 people, large clinical trials unit, and so we do a number of studies. Shown here is a slide provided to me by Gary Firestein, and basically this is a very simplistic depiction of the signaling pathways that occur after ligand binding to various receptors on the cell surface. And these signal transduction pathways take message to the nucleus, leading to gene transcription and upregulation of pro-inflammatory cytokines or anti-inflammatory cytokines or whatever. In the last 20 years, I've been working with a number of trials looking at each one of these parameters other than NF-kappa beta. It was interesting to hear about the NF-kappa beta trial in lupus. And part of this was we had these wonderful biologic therapies. As I mentioned, 1998, etanercept was approved, and infliximab was improved, and it dramatically made my life easier and our patients' outcomes much better. But these therapies are all parenteral, they're all expensive, so the holy grail would be, can we come up with a pill that will be as effective as those biologics? So we spent a great deal of time coming up with inhibitors, p38 MAP kinase. That was felt to be a crucial kinase in signal transduction, and is. And if we could inhibit that, potentially we would get better outcomes. All of these failed due to modest clinical efficacy and very similar toxicity profile of folliculitis, LFT elevations, and so forth. John Kay was the principal investigator on a MEK kinase, which failed in phase 2. We'll talk a little bit about 6, spleen tyrosine kinase. The Janus kinases finally achieved the hurdle, and there is one on the market, which we'll spend most of our talk discussing some of the data that we have, because I think in the future all of the new Janus kinases will be compared to that, looking for either better efficacy, different, maybe less frequent administration, so forth, and side effect profile. Very early data with BTK inhibitors, which was mentioned in the previous talk, and PI3K inhibitors. These are being looked at in rheumatoid arthritis, and there have been clinical trials ongoing, very early clinical trials. So this is a list I show. In yellow are all of these small molecule inhibitors for rheumatoid arthritis that we have studied that failed in rheumatoid arthritis. You'll note here apremilast, which did get approved for psoriatic arthritis with moderate clinical benefit as far as skin and joints, and a very nice safety profile. It's a PDE4 inhibitor, and it leads to down regulation of TNF-alpha and IL-1 production. There are still some small molecules looking at GM-CSF production, blocking the receptor or the molecule itself, and then, as I mentioned, the JAK inhibitors have successfully moved forward. So just a little bit of basic information about these kinases. Much of this work was done by John O'Shea at NIH and his colleagues over the last 20 years, elucidating these pathways. But there are 518 kinases which have been identified, and basically this is all involved in protein phosphorylation. So they transfer phosphate groups from ATP or GDP to free hydroxyl groups on amino acids. Most of these are serine and threonine kinases. There are 90 PTKs, or protein tyrosine kinases, that have been identified, and these are activated when ligands bind to type 1 and type 2 cytokine receptors. These activated intracellular PTKs phosphorylate tyrosine residues, resulting in peptide activation and signal transduction. There are receptor PTKs like platelet-derived growth factor, epidermal growth factor, and there are 32 non-receptor PTKs, of which JAK and SYK are part of. And again, another simplistic cartoon, but basically there are 4 JAKs, JAK 1, 2, and 3, and the Janus kinases, and TYK2. And the original target, at least by most, was felt that JAK 3 would be the most intriguing target. It's only seen in hematopoietic cells. And a number of the pro-inflammatory cytokines, IL-2, 15, 21, signal through JAK 3. JAK 3 always pairs with JAK 1 and the common gamma chain family receptor to transmit signal. JAK1 can also signal with JAK2, can also signal with TYK2, and so forth. And you can see the different cytokines. IL-6 signals through JAK1, JAK2, and JAK1 and TYK2, IL-12 and 23 through TYK2 and JAK2, and the various hormones like EPO, GM-CSF, thrombopoietin will signal through JAK2. Now, originally it was felt that going after JAK1 or JAK2 would not be a good thing to do because the knockout mice die. But indeed, that turned out that indeed you could go after JAK1 and JAK2, and much of that data came from the oncology groups. Again, by inhibiting this activation of these Janus kinases, we block upregulation of the STAT proteins, which are critical for signal transduction. So what do these inhibitors do? So basically, these PTKs bind to ATP and protein substrate in the cell. And they, again, lead to protein phosphorylation. So these PTK inhibitors get into that binding site and competitively inhibit that binding of ATP, which was said originally felt possibly could not occur because there's tons of ATP in each of these cells. There was also concerns that these PTK inhibitors would not be very selective. And if you inhibited multiple intracellular kinases, that you might really impact cellular signaling and lead to cell death. But again, oncology led the way. It's a lot easier to give drugs that you're concerned about to people who have a shortened survival. And now there's at least 13 PTK inhibitors approved in oncology. There have been a number of assays looking at the selectivity of these PTK inhibitors, and there's biochemical assays, there's enzymatic assays, there's cellular assays. And so you have to be very careful when you're looking at these results to try to compare apples to apples, looking at whether you're inhibiting only JAK1 preferentially or JAK3 or whatever. So this is a kinome map which has all of the kinases, and the tyrosine kinases are up here in the far left corner. And these are drugs that we're familiar with: imatinib, Gleevec, sunitinib, Sular, and erlotinib, which is used for lung cancer and pancreatic cancer. This is sort of a pan-kinase inhibitor. So you can see that originally the concern about selectivity has somewhat gone away because these PTK inhibitors, these tyrosine kinases, inhibit multiple tyrosine kinases as well as some off-target kinases. So this is tofacitinib, Xeljanz, which is now approved, and this is its kinome. And you can see that it certainly inhibits at a very low nanomolar concentration JAK3, JAK2, and it's been said that JAK is preferential for JAK3 and JAK1 over JAK2. The problem with this is that if you take the average level in the serum that you achieve with the doses we're giving at tofacitinib, you'll have an average of 90 nanomoles. So clearly this is as Peter mentioned yesterday, this is a pan-JAK inhibitor. So we're not, TYK2 may be escaped, but JAK1, JAK2, JAK3 are all inhibited. And the whole efforts in research at this point in the newer generations that are just entering the development are to come up with a more selective JAK inhibitor that might just get JAK3 or might get just JAK1. But even baricitinib, which is the next to come following tofacitinib, is really more of a pan-JAK inhibitor, although it's been promoted as a JAK1/2 inhibitor. And this was a study from the Pfizer group, their basic group in Boston, and they came up with this methodology looking at inhibitory capacity, what percent of inhibition of these signaling for these various cytokines. And they looked at tofacitinib and the dose that is approved in the States, baricitinib, the dose that's moving forward, 4 milligrams, and filgotinib, which is 200 milligrams, which is more of a selective JAK1 inhibitor. And what they showed is for most of these cytokines, this is a cellular assay, that there was similar inhibition of these cytokines, not a great deal of difference. There was some difference with IL-15. And one of the interesting things with tofacitinib, it's one of the few therapies that I know of that does inhibit NK cells. So it was interesting hearing that talk about NK cells and Sjogren's, and you wonder if there might be a role, because certainly you can see a significant drop in NK cells with tofacitinib that last for 6 to 8 weeks after discontinuation of the drug, and whether that has any role in the viral signature we see or whatever is not clear. So this is what happens when you give a JAK inhibitor, and this is a depiction for tofacitinib. Basically, JAK3 and JAK1 are not upregulated, not activated, so there's no activation of STAT and no transduction of signal. So intracellular blockage of signaling to the nucleus. So as we know, this drug was approved in 2013. It was approved by the US FDA for moderate to severe active rheumatoid arthritis, with inadequate response to one or more DMARDs, with some caution for use in the elderly and those with comorbidities. It can be used as monotherapy. It can co-administer with methotrexate or other non-biologic DMARDs, and only the 5 milligram dose was approved here. The 10 milligram dose was not approved, and as we know, the European Medicines Agency has not approved tofacitinib to date for concerns about toxicity as well as concerns over cost. So I'm just going to go through the clinical pharmacology of these therapies. It's well absorbed, has a very short half-life, about 3 to 4 hours. The Cmax is in 1 to 2 hours. It has a partial and intermittent suppression of JAKs. The question is, should we be suppressing these for 24 hours totally? There's some concern about total suppression and toxicity. This drug, like baricitinib, which will be the next to come, they're all teratogens. So clearly, these folks who want to pursue pregnancy should be off these drugs for probably at least 8 weeks. As I've said, the biologic effect of these drugs, even if the PK is short, the PD is 6 to 8 weeks. The clearance by multiple elimination pathways, both hepatic and renal. Again, with the dose that's approved, there's no requirement to lower the dose with moderately impaired renal or hepatic function. So there were a number of studies that were done in phase 2, and I just want to show you this data. You can see it was a very wide dose range. Let's see if I can get this working. Very wide dose range from 1 milligram twice a day all the way up to 30 milligrams a day. And with all of these Janus kinase inhibitors, we do see, with dose escalation, an improvement in ACR responses with a flattening of the curve, and the flattening of the curve occurred at about 10 milligrams BID, which is why the decision was made based on efficacy data as well as toxicity data to move into phase 3 with the 5 and 10 milligram BID dosages. It was a large program. 4,700 patients were recruited. There were 6 phase 3 clinical trials. It was predominantly an ex-US database that was accumulated. 30% of the patients came from Asia, 15% from Latin America. The disease duration was long in the majority of the trials, predominantly rheumatoid factor positive with high disease activity scores, and 2/3 were on oral corticosteroids. And actually, when you take a look at the baricitinib development program, and there'll finally be a number of presentations at EULAR, we think, well, the abstracts come out May 10th. On that database, it's also a huge database as well with very similar types of patients. Doing clinical trials has become incredibly hard in the States. For those of you who do clinical trials, you know this already. We have so many treatment options for our patients that it's hard to recruit, and the people we're recruiting for trials are either underinsured or don't have insurance. So pharma has had to go the rest of the world to find these patients to finish these trials in a reasonably acceptable period of time. So just to show you very quickly, one of the points that was made yesterday by Artie is that patients who are on placebo in these trials compared to 5 and 10 milligrams tofacitinib, and these were the DMARD incomplete responder trials, at 3 months, they were allowed to exit the trial if they had not had a greater than 20% improvement in swollen and tender joint count. So you lose a number of your placebo patients by the end of 3 months. So it's very hard to compare the efficacy and the safety, especially safety profile of these therapies to placebo when you have such a short duration. At 6 months, all patients exited the trial who were on placebo and went on to active therapy at that time. So they were all rescued either at month 3 or month 6. And in the 6-month trials, which were the TNF-IR study or the monotherapy trial, at the end of 3 months, all patients were escalated to active therapy. So it makes it very hard to do comparisons from safety, radiographic outcomes, and the radiographic outcomes are frequently extrapolated from the time they leave the study, so it gives you sort of a false impression of what's going on. So this is I'm just going to show you snippets of the data. This is all well been published, but this is the ACR 50 response across studies. These are the DMARD IR studies. This in the black is Humira. Adalimumab is the active comparator. This is the TNF-IR population, again, seeing that the responses are not as robust as the DMARD IR population. And then you have the monotherapy group, which showed very similar ACR 50 responses for the monotherapy group and allows us to use this as monotherapy. But again, as we're seeing now in a number of trials, this active comparator with the biologic is something we're seeing. It really gives you somewhat of a benchmark to how effective this therapy may be. Just wanted to show you this data, which basically is looking at the rapidity of the response. And you can see here that in the blue for TOFA 5 and the red for TOFA, purple for TOFA 10, that you can see that within just a month, there was a substantial number of patients who achieved ACR 20, and by 3 months, the response rate begins to plateau with these therapies. The folks on placebo who were rescued began to catch up, as you can see. So a very rapid response, generally within 2 to 4 weeks. And so my approach in clinical practice, if a patient hasn't had a response by 8 weeks, I don't think they're going to respond. We'll begin to talk about alternative therapies. And then clearly the bottom line is physical function and preventing disability. This is looking to hack the eye response. This is what we consider the minimally clinically important difference, and you can see for the 5 studies. And clearly tofa, as well as adalimumab, was very effective at improving physical function and disability. There was a lot of controversy at the FDA advisory committee meeting, as well as far as approval. This is looking at the radiographic data, and this is the mean change from baseline, the green being placebo, tofa 5 in blue, and tofa 10 in orange. And again, as Peter pointed out yesterday and others have pointed out, for some reason we are not seeing the X-ray progression we used to see in our patients on placebo. Very minimal change over 12 months. There were a number of sub-analyses done that showed that in patients who were at greater risk of progression of disease, that indeed blocking JAKs did prevent progression of disease. But based on this data, the X-ray progression labeling, prevention of progression, was not given. A subsequent study, after a subsequent study, was the START study, which I'm showing here very briefly. This was taking people with early rheumatoid arthritis and was a head-to-head comparison with methotrexate and tofa 5 milligrams or 10 milligrams, again, the BID dose, and followed for 12 months. And the co-primary endpoints were improvement in ACR70 response and the change in the modified total sharp score. And again, taken from the New England Journal paper, you can certainly see here that tofa at both doses was statistically superior to methotrexate. And in addition, looking at the radiographic changes, it was statistically superior to methotrexate, even though, again, the changes were very minimal. And this led to labeling for prevention of radiographic progression. Now, we don't start with tofacitinib in early rheumatoid arthritis because one of the things that did really concern us after its approval was the cost. It's a cost of $25,000 to $30,000 a year, where you start with methotrexate, select out that third of patients who do very well, and then go on to biologics or small molecules at that point. So let's talk about safety. Originally, we had 7,000 patient years of experience at the time of approval. We've published in Arthritis and Rheumatism last fall data with more like 8,000 to 9,000 patient years of exposure. We're working on a manuscript now with 12,000 patient years of exposure. We have limited data for the placebo group, as you can see down here, and for adalimumab for the reasons that I mentioned earlier. So these are the issues that you look at in these clinical trials. And I'll show you a composite graph. And generally, most of these things are comparable to what we see as far as the incidence of these events to the biologic therapies that we're quite comfortable with. TB was seen, not surprising, with the multiple cytokines that are inhibited. And again, this is data as of April 2012 from our manuscript. There have been 16 patients. The majority were pulmonary, but extrapulmonary disease was reported. Most of these were ex-U.S. Only one case was reported in the U.S. They were all over the world, as you would expect. When you take a look at the bottom here at the rates of TB, it was really not a great deal different from what you would expect to see with biologic therapies or with the rates in those particular countries. There was an unusual thing of opportunistic infections. We weren't really used to seeing many opportunistic infections reported in phase 3 clinical trial development programs. Certainly post-marketing, pharmacovigilance, we see these reported. But again, there were opportunistic infections reported, esophageal candidiasis, most of which were found by incidental finding in endoscopy. There were 3 PCP cases, one of who died. There were 4 cases of CMV viremia, and some cryptococcal pneumonia, 1 case of BK encephalopathy that recovered. So again, you wonder if the impact on the type 1 interferons and so forth that occur with these types of molecules or the NK cells might lead to a slight increased risk of these opportunistic infections, or was it a factor of the countries that these studies were conducted in? And that's what we don't know. The other outlier was this signal of Zoster, which was also seen with the drug in lupus, with blocking type 1 interferon. But here is the rate for tofa, 4.3 per 100 patient years. And here's the rate for adalimumab, 2.81 in the phase 3 clinical trials. And that's much higher even than the rate that we expect with TNF inhibitors, which usually the figure is about 1 in 100 or 1.5 in 100. So clearly there was a background increase in Zoster, but with this particular JAK inhibitor, there was a higher likelihood of Zoster seen, and certainly as part of our discussion with the patients the benefit risk discussion that we have when we start this therapy, and one of the reasons that we're trying to get our patients to be vaccinated before going on biologic therapies. Again, this is a composite looking at the data for TOFA in 5 milligrams in blue, in orange is 10 milligrams, compared to a number of other clinical trials looking at mortality, serious infections, malignancy, and so forth. You can read this on the bottom. And what you can see, except for herpes zoster, the frequency of these events is similar to what we expect to see with biologic therapies. Now, looking at the long-term extension data, it did look like there was a bump up in the serious infectious episode rate with the 10 milligram dose compared to the 5 milligram dose. There was, even though the confidence intervals overlap here, there was a slight tick up in lung cancer, and the zoster persisted. And because of that concern, being that this was a totally new mechanism of action, the first approval of a new therapy with this MOA, the FDA elected not to approve the 10 milligram dose for usage. Now, to me, I would have liked to have had the opportunity for the 10 milligram dose, because when you take a look at the clinical trial data, the higher threshold outcomes such as ACR 70, DAS remission, things of that nature, were better with the 10 milligram dose. And I think there are patients who will respond to 10. The problem with the development program, and I'm not sure baricitinib doesn't have this issue because they're coming forward with just the 4 milligram dose, is these folks never did the study taking 5 milligram incomplete responders and escalating to 10. So we don't know if those patients would respond better after failure on the lower dose. Laboratory changes can occur with these medicines. Stefan did a beautiful review of IL-6, and this inhibits IL-6. Clearly, IL-6 levels drop with tofacitinib. You see some modest LFT elevation, but it's generally been manageable, generally much more likely in patients on methotrexate. We do see this increase in lipids. Drop in neutrophils is sort of a biologic effect of the drug, but rare severe neutropenia is seen. And I'll just show you a couple of the data sets. So this is the percent increase in LDL. There was a nice study in Arthritis and Rheumatology a couple months ago from we were one of the investigative sites looking at cholesterol ester catabolic rate in controls in patients with RA, and it is increased in RA patients. When you give them tofacitinib, you decrease the cholesterol ester catabolism, so your levels of LDL and HDL go up. And that's what's seen here, about a 15% increase, and that's why we monitor after 2 or 3 months a lipid panel and treat these patients if their lipid levels go up. There was also some data from that study suggesting that the HDL particles, the LDL particles, were less atherogenic. That's above my pay grade, but that is certainly an interesting observation. Lymphopenia is another one of the black box warnings with this particular JAK inhibitor based on very small numbers, and certainly it was very disturbing data for me. I'm sure those of you who are in the clinic looking at your patients and you look at the white count and you go, they got 400, they have a very low lymphocyte count. They're on steroids. They have lupus. But the bottom line is, in this particular study, if you had lymphopenia with less than 500 lymphocytes noted, you had a very significant increase, 33%, 50%, for all 40% risk of 40% infection risk, compared to the background with normal lymphocyte counts with 2 to 3% infectious risk. So I'm still waiting for someone to go back and look in their clinic and do a study and look at the infectious risk from all our patients who have such low lymphocyte counts. But clearly, if you see this in patients on tofacitinib, I think you do need to either act on that by taking them off the drug or have a discussion with your patient. So let's talk about in the last couple of minutes what's coming. Baricitinib is the next one in the queue. And again, these drugs are being developed not only for rheumatoid arthritis. Tofacitinib has submitted their whole packet to the FDA for psoriasis. They are developing it in psoriatic arthritis. It will be developed in other diseases. As you know from surfing the web, it looks like it's a treatment for alopecia universalis. So there's some other things that are going to happen with these drugs that are going to be very interesting. But baricitinib is the next in the queue for RA. The Vertex JAK inhibitor, which was preferential JAK3/1 inhibitor, that development has been terminated. It was effective, but it had a little more LFT toxicity. The Galapagos specific JAK1 inhibitor has been purchased by AbbVie, and they're working with that. But they also have their own JAK1 inhibitor, which is different from the Galapagos compound, which is in clinical trials. Astellas has published data with their JAK3/1 inhibitor. There were 2 papers at the ACR meeting last year, one of which met their thresholds of ACR20 response and one which failed in a dose-ranging study to achieve its thresholds of response. And not clear exactly what's going to happen to this molecule. I think development is going to continue in Japan. I'm not sure it's going to continue here, but don't hold me to that because those things are in flux. So just to show you one slide on baricitinib, there really is very little in the public domain. We have the top-line results from press releases for their new studies, which show that it's working well at the 4 milligram dose. But in their dose-ranging study that Mark Genovese presented, looking at 1, 2, 4, and 8 milligrams given as a single daily dose in contrast to the BID dose, you could see there was substantial improvement, statistically significant difference in ACR responses, DAS response, CDAI, and so forth. And there was a sort of a dose response up until the 4 milligram dose, and above that there was no incremental improvement in these patients. So again, the 4 milligram dose is what was taken into phase 3 based on this data as well as toxicity data. There's a narrow window of benefit and risk with these drugs, and even the 8 milligram dose had a little more Zoster, a little more of this, and so they went with the 4 milligram dose. Thankfully, the folks preceding me talked to you about BTK, which is involved in B-cell receptor signaling, FC receptor signaling, spleen tyrosine kinase, PI3K, all again pathways that we can manipulate and look at and may be useful targets in rheumatoid arthritis. This is Mike Weinblatt provided me this slide. This is the Oscura data looking at fostamatinib, which is a spleen tyrosine kinase inhibitor, a huge development program. And basically, long story short, it did achieve its ACR20 responses, but did not show prevention of radiographic progression, therefore failed in its very large phase 3 study. This was a particularly toxic therapy as well. Significant number of cases of diarrhea, significant cases of hypertension, and transaminitis, probably due to off-target involvement. We also worked with another spleen tyrosine kinase inhibitor that Merck had, which basically we presented last year, and it too failed for toxicity. So PI3K inhibitors, just to let you know, there was a study that was just completed. All we have is the press release, delta gamma inhibitor, which was felt to be the primary target for the PI3K inhibitor. They failed to achieve their primary endpoint in a phase 2 dose-ranging study, and that development has been terminated. We're working with some other PI3K inhibitors in phase 1 development. We're working with several BTK inhibitors in phase 1 right now in rheumatoid arthritis, and we'll see what that bears. So, last slide. There's a narrow window of benefit to risk with the kinase inhibitors. We've learned that. For example, tofacitinib was looked at in the transplant population, somewhat higher dose in combination with mycophenolate, and clearly was too toxic as far as infection risk. So we need to find that sweet spot on the dose. Will long-term safety be acceptable? So far, short-term data is acceptable, at least through about 2 or 3 years of follow-up, post-marketing vigilance, and so forth. Will compliance with the BID dose be an issue? Doesn't seem to be at this point, but I'm not sure everyone needs 10 milligrams, and I'm not sure if my patients are telling me the truth, but the ones who are taking it seem to be doing well in general. The cost is an issue. Where does it fit in the treatment paradigm? I think like anything, when you're new to market, you're a new mechanism of action, you have physicians who are early adopters, you have physicians who are concerned and worried about side effects, and will use it late in the paradigm. We're still getting our hands around it. I use it in patients who are generally healthy, not the elderly, not patients on high-dose steroids, and we've had some reasonable success. I'm actually extremely excited about off-label uses of this drug, which I'm not going to talk about today. And so, will the subsequent kinase inhibitors have advantages over this drug? The only way we're going to find out, get our fellows to do a few comparative effectiveness studies. So we'll find out in the future if there is any significant difference by inhibiting different JAKs slightly better, or are these going to all be pan-JAKs? So thank you very much for your attention.